So today I'm going to be talking about my recent work over the last few years, considering the information-theoretic analysis of signal transduction. So as we're about to see, signal transduction is a very important molecular communication method in biological organisms. And by analyzing it from an information-theoretic perspective, the goal is to firstly understand how engineers could exploit this in molecular communication systems. And secondly, to help biologists understand the operation of biological systems a little better. Okay, so in the introduction, you heard about my textbook. I'd just like to put in a quick plug that if you want more information about molecular communication or my work, please feel free to read it. We also have a course, a free online course in molecular communication that's available at this URL, http://course.eckfordlab.org. So this is actually a course that I gave 2 years ago at an information theory school online. It was organized by one of the IITs in India. I'd also like to acknowledge my collaborators in all the work that you're about to hear about today, Peter Thomas and Ken Laparo at Case Western. And I'm always happy to hear from any researcher in this field, and please feel free to contact me by email. There's my email on the screen. So with that, what is signal transduction? So cells talk to each other using signal transduction. It's the main mechanism for communication amongst cells, both within your body, for bacteria, for basically any kind of cell, any kind of cell-cell communication is carried out by signal transduction. So the work on this goes back many, many years. Two of the principal researchers in this field were these two gentlemen on the screen right now, Alfred Gilman and Martin Rodbell. So their concept was that they realized that certain stimulants, such as adrenaline, had massive effects across the human body that couldn't simply be explained by the fact that they were widely different effects in widely different tissues. So they couldn't simply be explained by the fact that adrenaline could have a single effect. So what they realized was that adrenaline actually binds to various different receptors in various kinds of cells. And across the membrane of those cells, the signal of adrenaline is transduced in different ways, producing what are called G proteins that carry on different functions in different systems. So they realized that this was a transduction. It was taking one kind of message and transforming it into another kind of message. And for this insight, they won the 1994 Nobel Prize in Medicine. Rodbell in particular was inspired by electrical engineering. He thought that cells operated in terms of, he thought that the ways cells operated could be described in terms of circuits, in terms of like things like amplifiers, transducers, detectors, and things like that. And so he was, by being inspired by that, he went out and discovered signal transduction. So that's the origin of what we're going to talk about. So when I say that cells talk to each other using signal transduction, that's true in a very broad sense. So signal transduction controls basically every multicellular process in the body: cell growth, differentiation in the embryo, apoptosis, immune system regulation, metabolic control. Basically any multicellular mechanism within the human body is conducted by signal transduction. And in a very real sense, multicellular organisms can't exist without it. It's basically what makes you a human body instead of a collection of cells. So obviously this is a communication process. So it makes sense for communications engineers to think about how this might work. And so you might be thinking, well, this is a very complicated process. How can we as communications engineers even start to understand what's going on? So one of the insights, and perhaps if the only thing you take away from my talk is this, one of the insights that I'd like you to take away is that it's actually quite simple. Signal transduction is a Markov process. So if you know if you know anything about Markov chains, you don't need to know very much about the underlying biology in order to understand what's going on in signal transduction. So this turns out to be a very interesting theoretical insight that is actually very old. It was first realized probably 100 years ago. The mechanism is called mass action kinetics, and basically what this means is that reactions occur, like the timing of reactions at the microscale is timed using exponential random variables, which basically means that they're Poisson processes, which means that they are Markov processes. So it turns out with a little bit of understanding of stochastic processes, it's very easy to understand what's going on in signal transduction. It's just a Markov process. So to motivate this, we're going to be talking about two examples this afternoon. We are going to be talking first about cyclic adenosine monophosphate receptor, or CAMP, which is a ligand-gated receptor found in amoeba. The reason why we're going to talk about that one first is that it's the simplest. So this is a model organism for signal transduction, and it has the simplest possible receptor. And then we're going to talk about, as the second example, a slightly more complicated, but not significantly so, receptor called Channelrhodopsin. And that's a light-gated receptor found in algae. So this will also illustrate the versatility of signal transduction. It can respond to a variety of signals. The examples we're going to see are ligands and light. Okay, so let's start with the CAMP receptor. So CAMP governs organization in slime molds. So this figure on the left illustrates how it illustrates the life cycle of a slime mold or a Dictyostelid. So when resources are abundant, we start in the unicellular growth phase. So there's lots of food for the slime mold. It behaves like a single-celled organism. It just eats the food and it's very happy. Once if you look at the arrow associated with starvation within that box, once the food starts to run out, then the slime molds engage in a very different behavior. What they start to do is they start to come together. So moving counterclockwise from starvation, they start to come together into aggregation. So basically, the slime mold starts to behave as a multicellular organism now. And they go around through loose aggregate, tight aggregate, around the cycle until finally we get to fruiting body on the right. And that's the image on the right, or some fruiting bodies. So the idea is that at the very tip of the fruiting body are some spores, and those spores will survive the famine. And once food comes back, then it comes back around the cycle. It's CAMP that signals this behavior. So basically, once starvation starts to occur, the slime mold cells start to signal to each other using CAMP, and that's how they decide to firstly form an aggregate and exactly where that aggregate will form. And I hope you can see this animation. So on the this is an image that represents waves of CAMP that are traveling through the colony. So on the left, this is just an image of the colony. It doesn't seem terribly well differentiated. But on the right is a visualization. Actually, this is an experimental visualization. This is actual results that shows how CAMP is flowing through the colony. And you can see that it forms a wave. So there are these waves that move through the colony, and the center of those waves, that's where the aggregation will happen. So that's the mechanism that's going on. We're going to be studying this at a very, very, very microscopic level. So at the level of individual receptors, not necessarily at the level of the colony. But nonetheless, this is why it's interesting. So CAMP is what's called a ligand-gated channel. So ligand is just a fancy name for a molecule. It's basically the signal-bearing molecule. And the way in which cells communicate using ligand-gated channels is that cell one, the transmitter, if you like, emits a whole bunch of ligands or molecules. Those propagate to cell two, and cell two detects those molecules. So this is going from left to right, transmitter to receiver. from the perspective of the receiver, the receiving cell is covered with receptors. So these are CAMP receptors. The ligand that we're using to communicate here is CAMP. And what's going to happen is, so randomly through Brownian motion, these red circles, which represent the CAMP, will eventually intersect with the receptor. That receptor will bind to the ligand. While bound, it changes its configuration and it starts producing second messengers. So this is what I talked about at the beginning. This is the process of transducing the CAMP signal across the cell membrane. So second messengers are produced. While processing, there's a process in which the receptor has to clear itself until the point where it's ready to receive new ligands. And this process will happen all over the cell like so. Our second example is a little different. It's light-gated, which means that it doesn't respond to ligands, it doesn't respond to chemicals, but instead it responds to light. This receptor is called Channelrhodopsin, and in the wild, it's found in algae. So this electron micrograph on the right is the algae in which a certain Channelrhodopsin called Channelrhodopsin 2 is found. That's Chlamydomonas reinhardtii. This is again a model organism. Basically what happens is that a photon will hit the Channelrhodopsin receptor, which you can see depicted on the right, which again crosses the cell membrane. Once the photon hits Channelrhodopsin, the Channelrhodopsin receptor will open and admit an ion current through. I'd also like to point out Channelrhodopsin is very interesting, not only as a model organism, but also as a target for a technique called optogenetics that is used. It's been used over the last 10 years as a mechanism for control of individual neurons. And that's an entirely separate topic, but very, very interesting to read about. I don't have time to talk any more about it today, but it's quite interesting. So these are light-gated channels. So unlike the ligand-gated channels, now there are no ligands, or at least we don't care about them. So the environment, there are no molecules that we're reacting with. Instead, what's going to happen is that each of these receptors on the surface of the cell is going to get hit with a photon. Once it's hit with a photon, it opens, excuse me, and an ion current will pass through. So the ion current will change the chemistry within the cell and cause the cell to do certain things. So that actually replaces the second messenger here, is the ion current, which is causing changes within the cell. And again, this happens all over the surface of the cell. There's many, many receptors on the surface. Okay, so again, come back to our idea that signal transduction is a Markov process. So now we know a little bit about the physics of what's happening, either ligand-gated or light-gated, many other kinds of gating, voltage-gated, mechanostress-gated, many things that can trigger these things. But fundamentally, they're all chemical reactions that occur microscopically. And as a result, they're described by mass action kinetics, and therefore they can be described as Markov processes. So a continuous time Markov process can also be called a Poisson process, and receptor states in continuous time form a Poisson process. So as a CAMP example here, let's suppose we have a concentration C of ligands in the environment, and there on the right, we have our orange receptor. So what's going to happen? There's a certain rate at which, let me back up for a sec. The reaction, the process of binding a ligand to a receptor is a chemical reaction. That reaction will occur at a certain rate, and that rate will be a function of the concentration. Obviously, as the concentration gets bigger, then that rate goes up. So in fact, under the principles of mass action kinetics, this rate is linear in the concentration. So some constant times the concentration. So the process of binding is a reaction. But then now that it's bound, once a receptor is bound, it can't bind again until it clears itself. So there's a process by which it clears itself called unbinding. At the same time, it is producing second messengers within the cell. But that unbinding process is itself a chemical reaction. It occurs at rate beta. But what's important to note here is that while alpha is a function of the concentration of the ligand, beta is not. So this reverse process occurs at an independent rate. So you can depict this as a finite state machine like so. So the two states, we have a two-state finite state machine. The two states are labeled U for unbound. In other words, that's waiting for a ligand to bind, and B for bound, meaning that it's occupied, it's processing the ligand. So going from U to B, we have a rate of alpha of C. So that's a function of concentration. Going from B to U, we have a rate of beta. So that is independent of the concentration. So you can imagine how this would go. Imagine there is a huge amount of ligand in the environment. So that forward rate U to B goes very fast. So those reactions occur very quickly. So once you're in U, you go immediately to B. And then coming back occurs at a certain constant rate. So you'll end up in B a lot, basically. If the concentration is very small, then those forward reactions from U to B are very rare. So basically, you will stay in U for a relatively long time and only occasionally go to B. But again, the return will always happen at the same rate. So what we do in this kind of analysis is we form what's called a rate matrix. It looks like this. The rule with the rate matrix is that every row has to sum to 0, and that basically represents the net flux through a state. So the net flux out of U into B is alpha of C. That has to be balanced by a negative alpha of C here. Same thing in the second row. Going from B to U, the rate is beta. That has to be balanced by a negative beta over here. Channelrhodopsin works much the same way, except it's now a 3-state channel. So you can see why CAMP is the simplest. You can't get any simpler than a 2-state finite state machine. Channelrhodopsin is only slightly more complicated. It has 3 states. And just quickly, here's how it works. So once a photon is incident to the receptor, it opens, and that will clearly, the rate of that reaction will clearly be proportional to the intensity of the light. So brighter light means that reaction tends to occur more quickly because there's more photons to be incident to the receptor. Once it's open, ion current is flowing, and then eventually it will close. So there's a rate beta for the channel to go from what's called open to what's called degraded. So now the ion channel is closed, but the receptor is not sensitive to the environment. It's not ready to receive a new photon. So further conformational change has to happen before it's ready to receive a new photon. That occurs at rate gamma. And now once that has happened, now it comes back to a state called closed, and now it's ready to receive a photon again. So here's what it looks like as a finite state machine. We have 3 states: C for closed, O for open, D for degraded. Going from C to O, that's a function of the light intensity, alpha of C. Open to degraded occurs at an independent rate beta. Degraded to close occurs at an independent rate gamma. So you have to go through both open and degraded back to closed before it's ready to receive another photon. And again, you can write, just like we did before, you can write a rate matrix like so. Again, every row has to sum to 0 just to balance out the fluxes through the states. Note that some entries in the rate matrix here are 0. That means those transitions are impossible. So for instance, in the first row, going from C to D, going from C directly to D is impossible. The rate of that reaction is 0. But nonetheless, this is what the rate matrix looks like. And as a state diagram, it forms a ring. Okay, so these are Poisson processes. As communications engineers, we are normally accustomed to dealing with finite state or discrete time. Finite state, that's true, but discrete time Markov chain. And in order to do that, what we have to do is we have to take this continuous time Poisson rate matrix and transform it into a state transition matrix. So it turns out that's very simple. I'm going to give you a very simple way to do it. There are slightly more complicated ways to do it, but for sufficiently small time step, they're pretty close to equivalent. But the way to think about it is that basically the the rate matrix is a rate. It gives you changes in concentration as a function of time. So what you have to do is you have to multiply the rate matrix by some time step, delta t. And then it turns out that in order to make the probabilities work out, you just take I minus delta TR. We're going to make this even simpler. So because delta t is just a constant that adjusts your units of time, if we already set up R so that the rates are all with respect to a unit time step, then we can simply say the state transition matrix P is the identity matrix minus R. So what does that mean? In terms of CAMP, here was our rate matrix, right? So we had a row in which we had the transitions from unbound to bound, and then the second row is bound to unbound. If we just take the identity minus R to form P, this is what we get. And this looks exactly like a state transition matrix with each row now summing to 1, not 0. And this should be very familiar to you if you know anything about Markov chains. This looks exactly like a two-state Markov chain transition probability matrix, and this is exactly what we're going to use. So in terms of mathematical analysis, now you can imagine how this works. So again, with CAMP, here's our ligands binding to the receptor. Let's suppose we're signaling with 2 levels of concentration, high and low. So now my concentration is high, lots of ligands. In that case, I'm using this transition probability matrix with alpha substituted for alpha H representing high. So that'll be a high rate, high concentration, so alpha H will be big. On the other hand, if the concentration is low, again, I'm signaling binary. I have low concentration, high concentration. Now I just substitute in alpha L. So that'll be a low concentration. So that's the way to think about it. So in these state transition, in these Markov chains, the state transition matrix will actually change. It's time inhomogeneous because what we're doing is we're signaling and we are changing the rate at which that reaction happens. So at low concentration, that forward binding rate will be slow, and so therefore the Markov chain will have this state transition matrix that I have on the screen. On the other hand, when the reaction rate is high, the state transition matrix will have alpha H instead. Okay. So that might seem restrictive, but we actually have a result. I won't go through the proof, it's not difficult, but it turns out that we showed capacity is achieved with inputs only at the lowest possible and the highest possible input. So in other words, if you have a finite range of concentrations that are available to you, then the best thing to do is to signal with use a binary signaling scheme where you only use the lowest possible and highest possible concentration. Now that seems a little unnatural. You'd think that concentration doesn't behave like a binary variable. It doesn't go low and high like that. It turns out there's a little bit of experimental evidence that signal transduction systems might be trying to do this. So for example, in neural systems, it's known that release of neurotransmitters, like the adjacent neurons try to flood each other with neurotransmitters. So in other words, they're trying to force the concentration very high, and then they use an enzyme to break them down. In other words, forcing the concentration very low. So there's some evidence that this is actually done in nature. Okay, so in order to prove what the capacity of this kind of signal transduction process is, we've actually solved that problem. I'm going to go very quickly through the derivation for your interest. So the trick is, what we did was we have the Shannon capacity C, and what we did was we sandwiched it between CFB, which represents the capacity with feedback, and the IID capacity. So we didn't know, like the trick with Shannon capacity is you have to figure out what the input distribution is, and we didn't know what that is. So we basically said, well, it's easier to calculate feedback capacity and IID capacity, so we'll see if we can bound it. Feedback capacity looks like this. So that mutual information is now replaced by what's called the directed mutual information. And what you're doing is you're maximizing over what's called the set of causal conditional input distributions that look like this. Basically, the next input, xi, is dependent on all the previous inputs and all the previous outputs. So with feedback, you get to look at the state of the channel before you formulate what the next symbol you're going to signal is. So in other words, in order to decide whether you're going to do high or low, you get to see whether the receptor is bound or unbound. Okay, this does not seem very simple at this point, but there's a neat result from Jun Chen and Toby and the famous Toby Berger that says that for channels of the type that we're using, all you have to worry about to decide what the next state is you have to know whether the state is bound or unbound. So it only matters what the current state of the channel is. That's enough to achieve feedback capacity. So it turns out that's quite simplifying. So if all I need to know is whether the channel is bound or unbound, then if it's bound, it actually doesn't matter what symbol I use. The next state is not sensitive to that. In other words, the transition rate beta is not dependent on whatever I choose, whatever concentration I choose. So I can choose whatever I want, and it doesn't matter. It doesn't make a difference to the capacity. More formally, conditioned on being in the bound state, there's no information being transduced. The mutual information between at the next state, given that the current state is bound, is zero. Doesn't matter what I do. And that makes sense. It's insensitive, so it shouldn't matter what's going on when it's bound. So if I know it's bound, then it really doesn't matter what I do. What does that imply? So what that implies is that I can pick whatever I want. It doesn't matter. I can pick whatever I want for the input distribution when it's bound. So what I'm going to do is I'm going to pick the same as when it's unbound, which means that whenever I pick the optimal when it's unbound, that's when all the information is being transduced. I'm going to do the same thing when it's bound because it doesn't matter. But effectively what that means is that it's not dependent on the previous state. This implies that the feedback capacity is achieved by an IID input distribution, which means that the feedback capacity is the IID input distribution. And given the sandwiching at the top there, this means that the capacity, which is sandwiched between the two, must be equal to both. So that's the very quick proof of the theorem that the input capacity achieving distribution is IID for signal transduction. And in fact, what this means is that you can write down the capacity of signal transduction for CAMP in almost closed form. Here it is. It's also found in the paper that we wrote. The reference is at the bottom. And for channelrhodopsin, it's something quite similar. So basically, the numerator there is the information transduced. Given that you know that it's in the sensitive state, the denominator is the probability of being in the sensitive state. So it's quite simple. And in fact, you can generalize this to any receptor that can be described this way. And we have a simple formula described this way. I won't go into detail here, but you can find the details in the reference at the bottom. Okay, so this raises the natural question, what is the capacity? How fast can you signal? So for channelrhodopsin, what we did was we went to the literature and we grabbed some of these rate constants that have been established by experiment, and we plugged them into our formula and we tried to figure out what the capacity of channelrhodopsin is. And what we found was that for a single receptor, and remember that this is a single molecule, the amount of information that single molecule could transduce was approximately 66 bits per second. So in terms of engineered data rates like 5G or something like that, that might not seem very impressive, but 66 bits per second for a single molecule sitting on a surface of a cell is very impressive, especially when you think that a cell will contain thousands or maybe even millions of these receptors. So we took this result even further and we asked, well, okay, what happens when you do have lots of receptors? So each cell will, again, have thousands, maybe even up to 1 million receptors. And what we proved was that for n greater than 1 receptor, the capacity, if you have n receptors, scales linearly with the number of receptors. So if you have a capacity of 66 bits per second for 1 receptor, you have 1 million receptors, now you have a capacity potentially of 66 megabits per second. So this is getting actually quite serious as far as communication rates are concerned. And in some sense, what we're basically implementing here is a form of biological MIMO. So we have a huge number of potential receptors, each transducing the signal independently. and you can scale the data rate linearly in the number of receptors. So we think this is quite exciting, and we're actually doing some further research on this. Okay, so to conclude, what's next for this line of receptors? So there's lots of other receptors we can investigate. We're actually investigating a more complicated receptor called CFTR, which is involved in cystic fibrosis. We can combine this result with other techniques like artificial intelligence and other instruments to gain more information about what's going on at the receptor level. And ultimately, where we'd like to take this is we'd like to turn it into diagnostic and treatment tools. So that's the direction we're taking this right now. And with that, I think I'm just about out of time, so I will conclude. Thank you very much.